Hello everyone, welcome to Lab 10. Today we are going to discuss about blood and blood formation. So we'll be covering blood cells and how they are formed. So first let's have some summary questions. The first question, name the encircled structure. Here we have a section of a vessel. So when we see the lumen, it's irregular, so it looks like a vein, but it's not a vein. If it was a vein, veins have 3 tunics or 3 layers. Or you might think it's a venule. As you know, venules have an endothelium and connective tissue, but it's too large to be a venule. Venules are not this much large. And in addition, in arteries and veins in the lumen, it's not a must, but commonly we can see red blood cells. But this vessel lacks red blood cell. So it lacks red blood cell. The lumen is very large, and the wall consists of an endothelium. So this is a lymphatic vessel. The second question, what layer of the heart is this a section of? The question tells us this is a section from the heart, and here we can see longitudinal and transverse section of cardiac myocytes. So this is a section from the middle layer, which is myocardium. The third question, name the layer indicated by the brackets. Here we have cardiac myocytes, so this is the myocardium. And this could be either an endocardium or epicardium. And in this layer, we can see these large pale staining cells. These are Purkinje fibers, and Purkinje fibers are found in the endocardium. So this is endocardium. Question number 4. The arrow is pointing to the lumen of what structure? Be specific. Here we can see a section of 2 blood vessels. And this is a tricky question. When we see the lumen, it's irregular and collapsed. So you might think this is a kind of vein, some kind of vein. But when we see the tunics or the layers, it has a well-defined internal and external elastic lamina. This section is stained for elastic fibers. So you can see the internal and external elastic laminae. They are clearly defined and they appear dark or black. So as you know, veins, they don't have this well-demarcated internal and external elastic lamina. So this is a muscular or distributing artery. And when you see this one, in addition to the internal and external elastic laminae, in between, in the tunica media, we can see this elastic laminae. So this is an elastic artery, and this one is a muscular or distributing artery. And when you see the learning objectives of today's lab session, by the end of this lab session, you'll be able to identify the different types of mature blood cells. You'll be able to identify the stages of erythropoiesis, granulopoiesis, and thrombopoiesis, or the formation of platelets. So first, let's see the mature blood cells. So when we see a blood smear, the most abundant cells are the red blood cells. So these biconcave disc like eosinophilic cells are red blood cells. They are the most abundant ones. The second type of cells are white blood cells. And as you know it from lecture, we have 2 types of white blood cells. We have granulocytes and agranulocytes. So the granulocytes, they contain both primary and secondary granules, but the agranulocytes, they contain only primary granules. And we have 3 types of granulocytes. We have neutrophils, eosinophils, and basophils. When we see neutrophils, they are the most abundant ones, and their granules are neutral. That's why they are known as neutrophils. It's not either eosinophilic or basophilic, it's neutral. And the main distinguishing feature of neutrophils is the nuclei is multilobed. It contains 2 up to 5 lobes. And when you see eosinophils, so as their name indicates, the secondary granules, they are eosinophilic. So they appear red or dark pink. So the color of the granules is the same color as the red blood cells. and the nucleus is bilobed. And the third type of granulocytes are basophils. So basophils, as their name indicates, the secondary granules are basophilic. They appear blue. And like eosinophils, the nucleus is bilobed. And basophils are the rarest white blood cells. They are very rare. They are less than 1%. So if you have a hard time finding in blood smear, you can use the legend. And when you see the agranulocytes, we have monocytes and lymphocytes. Monocytes are the largest cells. They are very large in size, and they contain a C-shaped nucleus. And lymphocytes, they contain a very large round nucleus, which almost fills the cell. So they will have a very thin cytoplasm. And the third type of blood cells are platelets. Platelets are small sacs of cytoplasm. As you know, they are fragments from megakaryocytes and they contain different types of granules. Now let us see slides from our slide box. We have 2 slides of a blood smear. We have this, the MH033 blood smear. So this is an entire slide of a blood smear, as you see it. And on the left side, the cells are densely packed. And on the right side, the cells are less densely packed. So when we see the right side, the cells are densely packed, they are overlapping, so it's hard to distinguish or to identify the different types of blood cells on this side. We have another option of a blood smear, but if you decide to use this slide, use the right side, where the cells are not densely packed. So here on the right side, the cells are not densely packed, but even the higher magnification is not that much high. So I will be using the other blood smear slide, the 033 HR blood smear. This one has a very high magnification. So let's zoom in and look for the different types of blood cells in this smear. So these the most abundant cells. Eosinophilic cells are the red blood cells. And in between the red blood cells, these very small sacs of cytoplasm, which are fragments from megakaryocytes, are platelets. And this is a neutrophil. The granules are neutral, and as you see, the cell contains lobes, several lobes. It contains 2 up to 5 lobes. In this case, it contains, I think, 3 lobes. And this one is an eosinophil. As you see, the granules are eosinophilic. They have the same color as the red blood cell, and the nucleus is bilobed. Oh, we are lucky here. We have a basophil. So the granules are basophilic and the nucleus is bilobed. It's hard to see the nucleus because both the granules and the nucleus are basophilic, but the nucleus is bilobed. So these are basophils. This is another eosinophil. This is another neutrophil. And this one is a monocyte, a large cell. Let's look for another one which shows the nucleus clearly. And this is a lymphocyte. So as you see, it contains a large round nucleus and the cytoplasm is very thin. This is also a lymphocyte. I mean, this is also a monocyte, but it's covered by red blood cells. Let's look for another one. This is a lymphocyte. Yeah, this is the monocyte. As you see, it's a large cell and with a C-shaped nucleus. So look around and try to identify the different types of blood cells. If you have a hard time finding especially basophils because they are very rare, you can use the legend. Now let us see blood cell formation. First, let's see erythropoiesis, how red blood cells are formed. So the first stage in erythropoiesis is proerythroblast. So proerythroblast, it's a large cell with a large nucleus, and the nucleus contains two prominent nucleoli. And these nucleoli, they will produce ribosomes, and they will pump the ribosomes into the cytoplasm. So when the ribosomes are packed in the cytoplasm becomes basophilic. So now the proerythroblast, it becomes a basophilic erythroblast. So when you see the basophilic erythroblast, as you see, the cytoplasm is very basophilic due to the ribosomes, and it contains a large rounded nucleus. And when these ribosomes begin to produce protein, specifically hemoglobin, the cytoplasm will have eosinophilic patches due to the hemoglobin. Now the basophilic erythroblast, it becomes polychromatophilic erythroblast, multicolor. It will have, mainly it's basophilic, but it will have eosinophilic patches. But still, the nucleus is large, rounded, and it has a textured appearance. It's active. But when the nucleus becomes inactive and condensed, the polychromatophilic erythroblast, it becomes a normoblast. So when the nucleus is densely packed and inactive, when it's dark and homogeneous, the polychromatophilic, it becomes a normoblast. And when you see the cytoplasm, it's more of eosinophilic. And when the normoblast extrudes this nucleus, it becomes a reticulocyte. So when we see reticulocyte in a bone marrow section, in a bone marrow smear or section, they are less basophilic. Still, they have ribosomes, so they will appear less basophilic. So gradually, they will lose the remaining ribosomes, and they become a mature red blood cell or erythrocyte. So mature red blood cells, they are packed with hemoglobin. So now let's see a red blood cell development slide from our slide box. You will not be responsible to identify proerythroblast, so we'll start with basophilic erythroblast. So this is a bone marrow smear. Let's zoom in and see for... So here we have, let's zoom in. So this is a basophilic erythroblast. As you see, the cytoplasm is very basophilic due to the ribosomes, and the nucleus is large and textured because it's an active nucleus. And when these ribosomes begin to produce hemoglobin, we will have a polychromatophilic erythroblast. So as you see, the cytoplasm has eosinophilic patches. So this is a polychromatophilic erythroblast. And the nucleus is large and round, and it has a texture. And when this nucleus becomes inactive, when it becomes highly condensed, it becomes a normoblast. So here, as you see, the nucleus is dark and homogeneous, it's inactive. So this is a normoblast. And in addition, the cytoplasm is more eosinophilic. And when this normoblast extrudes the nucleus, it becomes a reticulocyte. So you can see it here. These are reticulocytes. And the reticulocyte, it contains ribosomes, so it's lightly basophilic. When it loses these ribosomes, it becomes very eosinophilic and it becomes a mature red blood cell. So look around. For instance, here we have a transition between polychromatophilic and normoblast. This is a normoblast. And these are reticulocytes. So look around and try to identify the different cells in the erythropoietic lineage. Now let us see granulopoiesis, so the formation of granular white blood cells. So the first stage in granulopoiesis is myeloblast. A myeloblast is a large cell with 3 up to 5 nucleoli, and it will be changed into promyelocyte into the next stage. And myeloblast and promyelocyte, they are common precursors for the 3 types of granular leukocytes, neutrophils, eosinophils, and basophils. So you'll not be responsible to identify myeloblast and promyelocyte. So let's start with myelocyte. So we have neutrophilic myelocyte, eosinophilic myelocyte, and basophilic myelocyte. So their difference is in the color of their granules: neutral, eosinophilic, and basophilic. And the other distinguishing feature of myelocyte is they contain a large round nucleus. So the nucleus is large and round in shape. So when the myelocyte is changed into a metamyelocyte, the nucleus begins to indent. So when the nucleus begins to indent, or when it's indented, the myelocyte, it becomes a metamyelocyte. So we'll have neutrophilic, eosinophilic, and basophilic metamyelocyte. And in the neutrophilic lineage, we have additional stage, which is known as band stage or band form. The nucleus, it becomes C-shaped, and it has equal width throughout its length. So neutrophilic lineage has one additional stage, which is the band form. And in the band form, the nucleus is C-shaped, and it has equal width throughout. It looks like a sausage. But in eosinophilic and basophilic, the metamyelocyte, it will be changed into a mature eosinophil and basophil. So now let's see these stages in a slide. So I will be using a neutrophil development slide. But in chapter 8 of the histology guide, we have different slides for each type of granulocytes. We have neutrophil development slide, eosinophil development, and basophil development slide. If you have a hard time finding these different cells in a different stage, you can use these separate slides and you can use the legend. But I'll be using the neutrophilic development slide. So let's zoom in and look for a neutrophilic lineage. So as you see, this is a neutrophilic myelocyte. The nucleus is round and the granules are neutral. So this is a neutrophilic myelocyte. And when the nucleus begins to indent, it becomes a metamyelocyte. So this is a metamyelocyte. And in the neutrophil development, we have additional stage, which is a band form. So this C-shaped nucleus, as you see it has equal width throughout its length. So this is the band form or band stage. And when this nucleus becomes lobulated or segmented, we will have a mature neutrophil. And here we have a eosinophilic metamyelocyte. as you see, the nucleus begins to indent. So this is eosinophilic metamyelocyte. As you see, the granules are eosinophilic. Let's look for basophilic and eosinophilic myelocyte. You can see it here. This is eosinophilic myelocyte. As you see, the nucleus is round and the granules are eosinophilic. Now let's look for basophilic myelocyte and metamyelocyte. Yeah, here we have an basophilic myelocyte. As you see, the granules are basophilic and the nucleus is round. And now let's look for a basophilic metamyelocyte. As you know, they are the rarest white blood cells, so it might be hard to find the basophilic lineage, so you can use the legend to find this basophilic lineage. So here we have an eosinophilic metamyelocyte. Here we have a metamyelocyte, a basophilic metamyelocyte. As you see, the nucleus is indented and the granules are basophilic. And here we have also an eosinophilic metamyelocyte. The nucleus is indented. So look around and try to identify the different stages in a different lineage. And if you have a hard time, as I have told you, you can use the different slides for each lineage. Now let us see thrombopoiesis, or the formation of platelets. So as you know, platelets are fragments from megakaryocytes. So megakaryocytes are very large cells, multi-lobed cells, and they are found adjacent to sinusoids or capillaries. So they have cytoplasmic extensions, and their cytoplasmic extension extends into the sinusoids. And the cytoplasmic extension fragments and gives rise to platelets. So let's see bone marrow section. This is a section, it's not a bone marrow smear, it's a section of bone marrow. So as you see, the cells are densely packed because it's a section. And you can see these large cells with multi-lobed nucleus. These are megakaryocytes. And here we have, this is a megakaryocyte, and as you see, the nucleus is multi-lobed. And they are found adjacent to sinusoids. Here we have a sinusoid, which is filled with red blood cells. And their cytoplasm extends into the sinusoid, and it fragments and gives rise to platelets. So that's all about today's lab session. We have covered these slides, study these slides, and try to identify the different types of mature blood cells and the different stages of this blood cell formation. And this is